second paper, or the second page or third page somewhere of that worksheet um, goes with that. There we go. Can get rid of the thing above. Just running my. So for this, let's see. What do I want to use? I usually talked about something recently in class, and so I'm going on with that. And here I'm just pulling something out of the air. Okay. Let's look at flower color in pea pea flowers. So let's do big P is equal to purple. Okay, and little p will equal white. So our purple is our dominant color. And let's go with our wrinkled versus smooth. Okay. Yeah, I need to do it. And I'm just going to leave that. You get the idea. Wrinkled, and then our little one, smooth. Okay. Some letters are easier for me to make than others. Okay. All right. And wow, I really got that out of line. Okay. So this gets messy. You draw your Punnett square first. Well, let's see. What I'm going to do is I'm going to do the most difficult, rolling up my sleeves, doing the most difficult of these. And that is where we are going to cross two heterozygous for both traits. That's how that's worded. Heterozygous for both traits individuals. Okay. Oh, I don't like that. Starting off bad. Okay. Try to draw this really, really straight here. Probably a template somewhere where I can do a box, but then it probably wouldn't work the way I want it to anyway. Well, okay, we're gonna be a little crooked. So I always draw this as an elongated rectangle instead of a square. And then you divide your rectangle in half, just like you do the square. Okay, so it looks kind of like square with the elongated square. And then you come back in and you divide each of those halves again. Ah, can I erase just that one? Yeah, yay. Okay, so now here's the tricky part of this, and this may be giving you some trouble on the worksheets. You have got to figure out what kind of alleles each of these parents can produce. Okay, what combinations are going to go in the egg or the sperm after meiosis happens. Now for this, our two parents are the same, so we only have to go through it once, but on the test, I could give you, and probably will, give you a combination where one parent has a different genotype than the other parent, in which case you've got to look at both of them. Okay, so, and on the very first page, at the top there, those first few questions are asking what kind of alleles can those individuals pass on. That's exactly what it's prepping you for. So we're gonna use this one for example, so I can just write out here at the side, real easy. This large P can get passed on, okay, kind of, or this little P can get passed on, along with either this large W or this little w, one or the other. 50-50 coin toss, right? So if this one gets passed on, then it can get passed on with either this or that. Again, a 50-50 coin toss. So that gives us two possibilities. That gives us, we can have that, or we can have that. So let's say this is the male, okay? And so that means the sperm can have either this or that. But we're not done, okay? Because the little p could get passed on. And I'm probably going to need to wind up curling the little p tail around like that in order to keep it away from looking like the capital. Okay, so I'm going to come down here, do that. We can have that, or we can have, I'm going to have to write it over the side because we're out of room. Okay. Because this could get passed on, but it could get passed on with this or with that. Foil, the first, the outside of the pairs. The first two of the pairs, the outside of the two pairs, the inside, and the last. So just like algebra, okay, foil, first, outside, inside, last works here as well to find our four possibilities of sperm that can be produced. And then over here, this is the female, possibilities of the egg, and it's going to be the same because these parents have the same genotype. So what you do then is up here, you have to write your allele pair combinations. Okay, and you're going to put your allele combinations for the other parent over here. These happen to be the same on this example, but if this parent had different alleles, a different genotype, then you would be writing different combinations down the side here. Okay, so don't forget that. That's you'll see it on your worksheet when you do your worksheet that there are different possibilities. Okay, this is just the most complex square, so I wanted to work it out for you. Has the most stuff in it. Okay, all right, so again, multiplication table style, just going to match them up. Okay, and at this point in class, I usually will take, I'm doing this on paper on the overhead. Okay, I will take it away. I will tell everybody to do it themselves, and I will do mine over the side. And then after a few minutes, I put this back up and I'll let them check themselves. So what you might want to do here is you might pause this. If you feel like you got the pattern that I'm doing here down, yeah, pause it, do it, and then come back and check what you've done. Okay, at the end. All right, and that's a good practice too. If you think you know what's going on, then go and test yourself, and then you can check yours with mine. And I'm just going to stop talking here for a little bit and finish this. Okay, so now hopefully here you can see why it's important that you keep the capital letter up front in each pair. Okay. And that's because what we're going to do next is we've got to go through here and figure out how many of the same type we have. Okay, so here I have one of these. Remember, the Punnett square is never the end of the story. The Punnett square itself is only the tool to find out your ratio. Okay, so I have one of those. I'm going to mark through that. Okay, probably should change this out here to, this is my list here, okay? Now, you can look for patterns. There's almost always a pattern. The pattern will need to be, we've got all the same stuff across the row. We've got same stuff in the columns. This row is the same as the third row. The second row is the same as the fourth row. First column is the same as the third column, something like that. What I was taught was to look at the diagonals, okay, across this first. And if you don't see a pattern in the diagonals, then start looking at rows and columns. And if you look, the scribble's really kind of hard to see it, but if you look, these two are the same. 2 capital P's, big W, little w. Okay. Now, if we look, we don't see that configuration anywhere else on this table in this square. So come over here and we're going to write 2 big P, big W. Well, you make that W. There we go. And little w. Okay. And we can cross those out. Okay. Big P, little p, big W. Well, that's not the same, but oh, look, this down here is. Okay, let's take those two. Okay, let's go back and look at this. Big P, little little w, little w. Nope. Okay, we kind of pass this halfway mark and everything becomes big P, little p, or little p. So that's our last big P, really. It's all by itself there. So one of those. Okay. All right. Big P, big W, little w. Oh, wow. 1, 2, 3, 4 of those. Do you see that? Okay. 4. Okay. Now, another thing too is that on the diagonal, sometimes this thing can like fold over. It's like a flip. So I can take the bottom of this and I can just flip it on the top side and see almost a mirror image of the same pattern, except that we're doing it with little p up front instead of big P, 2 big P's instead of homozygous dominant in our first allele pair, we've got homozygous recessive in our allele pair. Okay. For a lot of this, got 2 exceptions here. So normally I'd probably say, well, let's take these 2 here, but I'm going to actually start here in the middle with this one, and you'll see why in a second. And then we'll take these 2 here that are the same. These 2 here that are the same. Wow, those P's start to look like E's or something, don't they? Okay, I'm curving them like that. And 1, I'm hoping your screen is the same as mine and that the bottom of my screen is not cut off from your view. Okay, but I got 1, that last little square by itself, that it's homozygous recessive for both traits. Okay, so look at the number pattern we've created here. 1, 2, 2, 1, 4, 1, 2, 2, 1. That's why I took the middle inside there, so I'd have that repeat of 1, 2, 2, 1. This is a textbook standard pattern that you should memorize. Now, this is a list, and on the tests, I will ask you, if I ask you to write it out, I will ask you to write it as a ratio. And so the ratio is written, remember, not this way. Ratio is actually written dot, dot. Got to do dot, dot, okay? And I'm not going to continue to write all this out, but you would, okay? It goes on and on and on and on until you get this whole list put out into this format. The reason why you list first is because sometimes you have things here where you are you may miss one and you cut it back and it's like I may have put 3 here and then later went, oh wait, there's 1 there, and then you can look to see if you've got any repeats in this list and you can consolidate. So this would be the answer right down here, carried on out to the end. This would be the answer of the ratio. If I ask you to find the genotypic ratio, I'll write that word out. Ask my daughter to silence my phone for me because one of my groups is talking a lot right now. Okay, so that's your genotypic ratio. But that's probably not done with this question because I will probably also ask you to give me a phenotypic ratio. And on the short answer portion of the standard test, I do like 2 of these. One of them's bonus, one's not, but they're worth about 13 points total because this right here is worth 9. So here's, if you count down your side, 1, 2, 3, 4, 5, 6, 7, 8, 9. So just getting this genotypic ratio all written out correct is worth 9 points. And then the next step is a phenotypic ratio, and that one's worth 4 points. So let me show you how that happens. I don't want to clear the whole thing. I want to leave my list over here because I'm going to use my list, but I want to clear all of this stuff here out. It's probably bigger, like I said, thicker, better eraser, faster way of doing this. Don't know, but anyway, learning as I go, we'll get this to work. Okay, I'm going to change colors. Going to go to green for gene. Yeah, genotype. No, phenotype. This is a phenotypic ratio. I think green will be good and distinguishable here, though. Okay, so phenotype is where we write out the description. So, for example, change my color, but I didn't get a mark. Okay. Wow, big dash. Okay, so this is purple and wrinkled. And I'm going to abbreviate. Okay. Probably start abbreviating more. Here we go again. We have, okay, this was easy. 2 dominants, right? 2 dominants. Now we have 2 dominants, so dominant recessive, but that dominant's going to mask that. So we're going to have purple and we're going to have wrinkled again. And again here, because this little recessive is going to be masked by that dominant purple. Okay. And here, oh, we have a difference. This is purple. but this is smooth. Okay. Purple wrinkled. Okay. White, both recessive. And wrinkled. Purple and smooth. It's our first occurrence of smooth. Oh, it's not right up here. I said with purple, but it's not. Okay. White and wrinkled again. So I'm going to abbreviate that. Okay. And then our one homozygous recessive for both traits is white and smooth. I'll abbreviate that one too. Okay, now notice here how we have, again, a lot of ones that are the same when we had different genotypes, but those genotypes gave the same phenotypes. Okay, so what we do, because all the same color, now I'm going to change. What we're going to do is we're going to look at how many of the same ones we have. So I have one purple wrinkled, two purple wrinkled, two purple wrinkled. So that's 1 and 2 is 3 and 2 is 5. And do I have any more purple wrinkled? I do. Okay, so, but they're got to skip down here to find them. So this is again where it's good to kind of cross off as you go, so you don't double count anything, and you also don't miss anything, because that's where students make the most errors on the test, is they miss something or they double count something. Okay, so I've got these right here is also purple wrinkled, so that brings you up to 9, and that's all my purple wrinkled. So making my phenotypic ratio. Okay, the next one on the list is purple smooth. I have one of those and one of those down here. So I'm going to cross that off. 1 and 2 is 3. I don't see any others. Then I have wrinkled white. I mean, yeah, white wrinkled. Okay, let me keep my traits in the same order. It's always a good idea. Keep your traits in the same order. Don't mix them up like that. Okay, so I have 1 white wrinkle and 2 white wrinkle down here. So that'll be 3. And then I have my last white smooth all by itself. So that's your phenotypic ratio. And for a cross like I had before I erased it as a heterozygous for both traits, both parents heterozygous for both traits. This is a pattern you're always going to get. It's a textbook ratio for that goes along with this. So we had the 1, 2, 2, 1, 4, 1, 2, 2, 1 genotypic ratio. For the phenotypic ratio, you get 9, 3, 3, 1 always. So for the 9, you have the 2 dominant traits together for both traits. Here you have one dominant with the other recessive, okay, and then the other dominant with the other recessive or recessive dominant. They swap, okay, as to dominant recessive pairs. And then here is the recessive for both traits, okay. So I'm going to end this here because I think that's enough for one, and I'll see you back for more later.